Carbohydrate catabolism is going to be a very common process inside cells, and this process is going to take carbohydrates or sugar molecules, carefully break them open so that we can get the energy out of them. So here's a look at the major processes that are used to break down these carbohydrates. We have two main pathways. One of them is called respiration or cellular respiration, and then the other one is called fermentation. These processes are going to be the primary means of providing energy for the cell, and then that energy that is provided is going to be used for anabolic reactions, which are the reactions we would use to build things inside of our cells. Regardless of which one of these pathways we are actually running, both of them do have pretty much the exact same starting point. Both of them are going to start with glycolysis, and so we'll talk about that glycolysis process first. And glycolysis is just going to begin the breakdown process. So we'll start with the glucose and then we'll start to break it down. Now you can get into a lot of complexity when you do start talking about the steps of glycolysis. So we are going to keep it on a simple level here. Glycolysis, just the word, if we break it down, means splitting sugar. And that is what we're going to do, although the overall process really involves about 10 different steps. We're going to have two halves to the process. We will have an energy investment phase where we actually have to spend some energy that we already have and invest it so that we can run the second half, which is the energy payoff phase. So by the second half, we're going to start to get some return on our investment. So if we look at the energy investment phase, we are going to start with a glucose molecule, which is a relatively small carbohydrate. It's just 6 carbons in size. And we're going to have to use some ATP to actually make this molecule more likely to split open, which is what we're trying to do here. So notice that we've got some ATP actually going into this process. So we see the arrow going in, which means we're using it. And then once we use the ATP, we will be able to break open and split the glucose in half. So we will be left with 2 partially broken down carbohydrates. And there's still a lot of energy left in those, so we're just getting started at this point. At that point in time, we have invested 2 ATPs, so we're actually at a negative value energy-wise if we were to stop there. But we're not going to stop there. We're going to take those partially broken carbohydrates and we're going to move them up to the other side so that we can continue to break them down and get the energy out of them. So the first thing that we're going to do on the energy payoff phase is we are going to begin to collect some of the high energy valuable electrons that those molecules contain. And the electron carrier that's going to come in and actually collect those electrons is called NAD+, and when it gains those electrons, we change the name to NADH. So NADH would be the reduced form that's holding on to those electrons. We will also be making some ATP. So notice that we see some ATP coming out of this reaction as a product. And then at the very end of it, we will have our broken open glucose. We have 2 3-carbon sugars, which are called pyruvate. There's still a lot of energy left in those pyruvate molecules, and that's what we will continue to break down if we are talking about cellular respiration or if we're talking about fermentation. If we were to tally up our payoff on the energy payoff phase side, we have made 4 ATPs, and we also have those 2 electron carriers, NADH, that are holding on to the valuable electrons. So if you take that number or those numbers and you subtract off our initial investment, we are at a positive at this point. Our net or our profit is going to be that we have 2 ATPs and we also have these 2 valuable electron carriers. Well, this is what we start with, glycolysis, regardless of how we're going to actually break down the sugar ultimately. If we look at cellular respiration is the most common way of continuing this process. With cellular respiration, it is going to completely break down the organic molecule, glucose in our example, and it's going to completely yield carbon dioxide from that. This is the overall equation of cellular respiration. We start with the glucose. We're going to have to use oxygen from the atmosphere, and then we will be ultimately producing water, carbon dioxide, and then the part that we're interested in is the energy. And then once that energy is released, we can take that energy and we can do things like make ATP with it. We do start with glycolysis, but after glycolysis, we're going to take the pyruvate, those little sugars that we've made, and we're going to continue to process them. We will have the synthesis of acetyl-CoA, then we'll have the Krebs cycle, and then we will finish it up with the electron transport chain. So if we look at these individual stages now, if you look up here at the top, we have pyruvate or pyruvic acid. It's the same thing. And notice that we are going to right away remove some carbon dioxide from this molecule. So you can see that being produced right here. That is a waste product, and that will be released, and it can be basically released right to the atmosphere. Then there are a couple more steps. We will see some collecting of electrons again by an electron carrier. So we will produce this valuable NADH down here, which is the reduced form that's holding on to those valuable electrons. And then at the bottom, you can see that we do have acetyl-CoA. That is the ending point of this particular process. So we have glycolysis, and then we're going to convert that pyruvate that we get into acetyl-CoA. We'll collect a few more electrons, which again is in our electron carrier. And then the acetyl-CoA that we make is going to be used for the next stage, which is the Krebs cycle. Here we have the Krebs cycle. The Krebs cycle, we have 8 steps listed on here. It is a complex cycle. There's a lot of detail we could go into here, but again, we're going to keep it at a simple level. Just a few things I want to point out about the Krebs cycle to you is that we will continue to remove and release carbon dioxide. So if we look at this, we've got carbon dioxide coming out here and here. Those, again, are waste molecules, so we would release those back to the atmosphere. And then other things that we're interested in here on this stage, or in this stage, specifically, we've got valuable electrons that we've collected in NADH molecules. So we've got several of those being produced. And we also have another electron carrier participating down here, so we can see FADH2. That one is also hanging on to some valuable high energy electrons. And then one last thing that we're producing in this stage, which is very important, is we do get some ATP produced down here at the bottom. So the citric acid cycle, or the Krebs cycle, it is oftentimes called, does finish breaking down the sugar, in that it finishes releasing the carbon dioxides, so breaking them down and getting them back to the atmosphere. We finish collecting the electrons that we're interested in, and we also make some ATP. And when we finish this stage, we are left with electrons. That's what we have to actually get more energy out of, because these are going to be really high energy electrons. There's a lot of energy still there that we can actually harvest or collect to make the ATP that we do want to make to run our other reactions. So we do have electrons sitting in multiple NADH molecules that have been produced, and we also have some that is in FADH2. So that's going to be our starting points for the next stage. This last stage is the electron transport chain. And the electron transport chain, I like to think of it as an assembly line, because at the very beginning of the assembly line, and you can see it right here, we have NADH, and that's the ones that we produced earlier. They are going to come into the first person or the first member of the electron transport chain, and they're going to pass those electrons in. And when they pass those electrons to another molecule, that's a redox reaction. So the electron transport chain is a lot of redox reactions. So that's a lot of passing electrons from one molecule to another. Every time a molecule loses an electron, we call that oxidation. Every time one gains the electron, we call that reduction. And we're going to be passing those electrons from one to the next to the next. And every time we pass them, a little bit of energy is going to be released from those electrons. So the point of this electron transport chain, or this assembly line, is to basically slowly drain the energy out of the electrons. And then as we drain that energy out in a very slow, controlled fashion, the cell is going to be able to harvest and collect that energy and use it for something good, like making ATP. Notice that at the very bottom of this assembly line, we are going to have a final electron acceptor. And this final electron acceptor that we do have is going to be oxygen. That is the oxygen that's present in the atmosphere. That is the reason why most organisms actually require oxygen in order to survive, and that's because oxygen needs to be here ready to accept those electrons after the energy has been drained from them. So that's the electron transport chain, and we have now released all of the energy from these electrons that we collected along the way. So energy's been released. Now we want to be able to do something with that energy so that we can really pack it away and use it later when we do have reactions like anabolic reactions that require it. All right, so this is kind of putting everything together for us. This membrane right here is going to have the electron transport chain actually embedded in it. So that is our assembly line. So all of these molecules that you see, the orange ones and the purple ones, that have all the arrows kind of passing between them, those are the assembly line members or the electron transport chain. And notice that up here at the beginning of it, we have our electron carriers, which we produced earlier, that are holding on to those valuable electrons, and they're basically passing the electrons into the first member. That molecule or that individual passes them from that one to the next, and then we go to the next and the next and the next. So as the electrons are being passed along, and as that energy is being slowly released from them along the way, that energy that's released is going to be used to concentrate hydrogen ions on one side of the membrane. So we'll end up with one side of the membrane that has lots of hydrogens on it, and then we'll have another side that does not have as many on it. And that difference in the hydrogen ions is what we call a concentration gradient, and the hydrogens want to flow back through to the other side. So we're going to take advantage of that, and we're going to use a special enzyme that can allow the hydrogens to flow back through and make ATP at the same time. And that is the enzyme that we're seeing over here. It's going to be making ATP. So let's look a little closer at that process of making ATP with the energy that has been released by the electron transport chain. So when we make ATP using this enzyme, it does have the name chemiosmosis. So that's the process. This is going to be an energy coupling mechanism, basically utilizing the desire of the hydrogens to want to flow back across the membrane. And we're going to use that energy to make ATP, which is going to be perfect for running lots of reactions inside the cell. We have a special enzyme that does this, and this enzyme's name is ATP synthase. So ATP synthase kind of works like a water wheel would work, in that it's going to allow the hydrogens to flow back across the membrane, and when they flow back across, it has this special wheel or rotor that is going to be turning as they move across. And so that turning motion is going to be used to put together ADP and phosphate, which is essentially charging the ATP molecule up. So we're going to use the energy in that hydrogen gradient to make ATP or recharge the ATP battery. So that's the cellular respiration side. Cellular respiration is going to completely break down the sugar so that we get as much ATP at the end as we possibly can. That process requires the presence of some oxygen because oxygen had to be there at the end of the assembly line to accept those electrons. So that was definitely part of it. The other way of breaking down carbohydrates is called fermentation. Fermentation is going to start with glycolysis just like cellular respiration did, but the big difference here is that fermentation is only going to partially break down the sugar. And since it's only partially breaking down the sugar, we will only partially get the energy out of it. So overall, it is going to be a much less efficient process than the respiration side. As far as the details of it go, again, we start with glycolysis. We produce the pyruvate or pyruvic acid, and we'll get a little bit of energy or ATP made along the way. Then we will have a few more steps that take place just to get the electrons out of the electron carrier. or regenerate the NAD plus is how we see it. So notice that we start with NAD plus up here and then we have to get it back. We have to recycle that constantly. Fermentation does not produce a whole lot of energy, but it does produce some, and for some simple organisms, that is enough for them. It is also something that is used commercially for a number of different products that we rely on every day. If you look here, fermentation products include Swiss cheese, other cheeses and yogurts and soy sauce. We have alcoholic products like wine and beer. And then we also have some that produce some acetone and some isopropanol. So we have products that are built on those molecules. So fermentation, it's very commercially important to us, and the ability of microbes to perform that process has been taken advantage of in a number of cases to give us the commercial products that you do see here below.